Ladies and gentlemen, please welcome to the stage Professor Linda Gale Becker, President of the International AIDS Society and International Scientific Chair of IAS 2017, to announce this year's collaborative initiative for pediatric HIV education and research, Cipher grant and fellowship programs.
Good morning, everyone, and it's my great pleasure today to make these awards. The Cipher grant program awards 2-year research grants of up to $150,000 to early stage researchers for projects that address priority research gaps in pediatric and adolescent HIV in resource-limited settings. Thanks to the continuous and very generous support of ViiV Healthcare, Cipher founding sponsor, and more recently an added partner of Janssen, it is with great pleasure that I award the following outstanding research projects in pediatric and adolescent HIV. The objectives of this program, as you see, are supporting early stage investigators, targeting pediatric HIV research, and the grants are for up to 2 years, $150,000. So our first awardee is Amy Louise Slowgrove of South Africa, who will be investigating, does initiation of antiretroviral therapy prior to immune suppression protect against maternal hypertensive disorders in pregnancy and associated adverse birth outcomes in HIV-infected women. Congratulations, Amy. Our next winner is Andrew Walker McCrary of the United States, Duke University. He will be assessing the indicators of cardiac function in children living with HIV. Well done, Andrew. Third, Uta Fucht of South Africa, University of Pretoria. She'll be assessing factors impacting on fetal and infant immunity and growth in HIV-negative and HIV and antiretroviral exposed uninfected children. Uta. And finally, Wippaporn Natalie Songtweeson of Thailand will be looking at youth-focused strategies to promote adherence to pre-exposure prophylaxis among youth at risk for HIV in Thailand. Congratulations. You want me to be there? No. Very good. Well done, everyone. Thank you very much. I'm pleased to say there's more. And as we continue to nurture our Cipher alumni, many of whom are presenting research very excitingly at this conference, we are pleased to announce the launch of a new round of pediatric and adolescent research grants. Applications open in October 2017. So please make sure you get your applications in early. This has become really popular and I think a fantastic effort. Thank you again to our sponsors. Now, building and supporting global research leadership in the response to pediatric HIV is the focus of Cipher's new Growing the Leaders of Tomorrow Fellowship program. The fellowships strengthen pediatric HIV clinical research capacity in sub-Saharan Africa by providing promising early-stage investigators with 2-year, $70,000 research fellowships, and by supporting their connections with internationally renowned mentors to help guide their investigation. So this is a natural progression of the Cipher Research Award Program. Again, thanks to the wonderful support of ViV Healthcare, it is with immense pleasure that I award the following outstanding research projects in our first round of this Growing the Leaders of Tomorrow. And the first goes to Audrey Becker, who is not any relationship to me. Working together with Prof. Mark Cotton, she will study the pharmacokinetic and safety characteristics of nevirapine and lopinavir-ritonavir in HIV-exposed and infected low birth weight infants in South Africa. Audrey. And secondly, my pleasure to announce Jane Namangolwa Mutanga of Zambia, who will be looking at long-term health outcomes and survival of perinatal HIV-exposed children in Zambia, designing a comprehensive model of care for long-term follow-up. Congratulations. Thank you, everyone.
To introduce our first plenary speaker, please welcome Professor Françoise Barré-Sinoussi. Françoise Barré-Sinoussi has been involved in retrovirology research since the early 1970s and has been recognized for her contributions to HIV and AIDS research, in particular, the discovery of HIV in 1983. Until 2015, as research director at the National Institute for Health and Medical Research, INSERM, and professor at the Institut Pasteur in France, she led research programs on HIV and AIDS pathogenesis, specifically on mechanisms required to control HIV SIV infection and harmful T-cell activation induced in response to HIV SIV infection. She serves as honorary president of the Institut Pasteur International Network and of the Virology Department of the Institut Pasteur in Paris. She was elected president of the IAS between 2012 and 2014. And is still member and chair of a number of international scientific advisory panels and boards. She also received more than 40 national and international awards and honors, including the 2008 Nobel Prize in Medicine for her contributions to HIV and AIDS.
Good morning, everybody. It's my great pleasure to introduce Paul Bienatz. Paul is the head of the Laboratory of Retrovirology at the Rockefeller University and investigator at the Howard Hughes Medical Institute. His work has provided key insight into the fundamental mechanism by which HIV replicates and subverts the function of the cells that it infects. Paul's studies have focused particularly on retrovirus particle genesis and on the discovery and characterization of host cells, genes, and proteins that have antiretroviral activity. His group and collaborators have exploited this basic finding to generate new animal models to study intervention in HIV infection and AIDS. As well as a particular interest in HIV variant genesis and antiretroviral particles, he also has a broad interest in molecular biology of retroviruses, Paul pioneered the field at paleovirology, and over his career, he has contributed to our understanding of retrovirus entry, post-entry events, and transcription. Paul, please.
Well, thank you very much. Thank you very much, Francois, for that introduction. It's really an honor to be introduced by you. So my talk today will be in 3 parts. The first part will be a biology lesson, where I tell you about restriction factors, what they are, what they do. Then I'll talk a little bit about how we might apply this knowledge to do useful things. And then the third part will be aimed squarely at the basic scientists in the audience, where I'll talk about some new work that we've recent discoveries. Okay, so what are restriction factors? Restriction factors are antiviral proteins. I'll use those 2 terms interchangeably. They mean the same thing to me. These are proteins that we make that directly or indirectly inhibit particular steps in the virus replication cycle. Unlike your adaptive immune systems, they are sometimes constitutively expressed, ready to go from the moment you see a virus. Sometimes they're interferon-inducible, can be mobilized within a few hours. As I'll illustrate, they have very diverse mechanisms of action. Some of them have activity against a broad range of viruses. Some are specific for particular viruses. They are invariant, unlike your adaptive immune systems, within an individual host. Nevertheless, they are rapidly evolving, so between species there is a high degree of variability, and this often underlies what we call host range restrictions. So if we think about how these proteins work in the context of the replication cycle of HIV, the antiviral proteins can be broadly divided into 2 classes. One class act in already infected cells to reduce the yield or the quality of the viruses that an already infected cell makes. A second class works in candidate target cells, essentially to reduce the susceptibility of that cell to infection by virus particles. So I'm going to take you through a few examples of these proteins just to illustrate the diversity of the mechanisms by which they work. My favorite is a protein called tetherin. Tetherin is a rod-shaped protein that sits on the surface of cells. It has 2 membrane anchors, and as virus particles bud through the surface of that infected cell, the tetherin protein itself partitions between the membrane of the infected cell and the membrane of the virus as it buds, thereby generating a protein tether that traps viruses on the surface of infected cells. And as you can see in these electron micrographs, the degree to which virus particles are restrained from going off to find other cells to infect can be very dramatic. Another class of antiretroviral proteins is a family of proteins discovered by the Malin group, the ApoBec3 proteins. These are, in effect, retroviral poisons. They act in the cytoplasm of infected cells, and they infiltrate retrovirus particles as they're budding from the surface of cells, and they do so in a very non-specific way. They recognize the RNA genome of the virus, and you can see on the right of this slide a newly obtained structure of the ApoBec3H protein done by Jen Bone and Janet Smith, our collaborators, which shows how the ApoBec3H protein recognizes short duplexes in RNA, but of no defined sequence. That enables ApoBec3 proteins to target a very broad range of viral RNA molecules. Once they're inside the viral particle, they ride along with it until it infects a new cell, and then they go to work during the reverse transcription step of virus replication, both inhibiting reverse transcription and causing a massive degree of mutation. These proteins are cytidine deaminases. They change the nucleotide sequence of the virus to a massive degree, causing it unable to be able to propagate. So the second class of antiviral proteins that acts in target cells includes molecules such as TRIM5, the poster child for this class of antiretroviral proteins, What TRIM5 does is work in the cytoplasm of a target cell. It recognizes the incoming viral capsid. You can see that the capsid itself is made of a hexameric lattice, and the TRIM5 also forms a lattice that's templated by the capsid and then appears to engulf the incoming viral capsid, ultimately leading to it and its contents being destroyed and the virus replication being halted. Another capsid-targeting antiviral protein exists in the cytoplasm of interferon-treated target cells. This is a protein called MX2. Like TRIM5, it recognizes the viral capsid, but does so at a later stage. It patrols nuclear pores and blocks specific nuclear import pathways that the virus particularly likes to use to gain access to the nucleus of the target cell. In so doing, it blocks nuclear import and infection. So these are just a few examples among the more prominent examples of antiretroviral proteins that have been discovered in recent years. This is an incredibly rich and fertile area of basic HIV research, and that not a year passes without a new antiretroviral activity being discovered that's encoded within our host cells, some of which are listed here. Now, one might be tempted to ask, confronted with this extremely hostile environment, how does the virus even replicate at all? And of course, evolution has endowed the HIV-1 and essentially all viruses that infect hosts with a number of countermeasures. And in particular, in the case of HIV, the so-called accessory genes, a major raison d'etre for these genes is to act as antagonists of the antiviral proteins that I have just told you about. For example, the ApoBec3 proteins are bound by a viral protein VIF that then tells the ApoBec protein to be destroyed by linking it to a cellular degradation pathway. The tetherin protein that I spoke about a few minutes ago is bound directly by the HIV-1 VPU protein or the SIV NEF protein, and that removes tetherin from its site of action. In the case of the capsid targeting restriction factors, these are evaded by the virus simply by changing the sequence of the capsid protein that is recognized by the antiviral proteins. So that gives you a sort of a primer about what antiviral proteins exist and how they work. How might we apply this knowledge to do something useful? Now, one thing you may notice about them, at least I hope you will have noticed, is that many of them target the virus in a way that's very difficult for the virus to escape from. They generally don't target the virus in sequence-specific ways, in ways that allow the virus to evolve resistance. Rather, the virus has taken the much more difficult evolutionary step of synthesizing new biological activities. And so, for example, developing drugs that would inhibit these interactions would be predicted to be potent antiretroviral compounds. That idea is yet to be realized in practical terms, although there are efforts in particular to develop antagonists of the HIV-1 VIF protein. There's only one viral protein that nature has chosen to target specifically by antiretrovirals, and that is the capsid protein of the virus, which is targeted by TRIM5 and by MX2, and in other retroviruses there are additional capsid targeting proteins. Now, why might that be the case? It turns out that capsid is a particularly difficult protein to mutate without running into profound fitness defects. And so this is an experiment done by a student of mine a few years ago, where what Susanna Reen did was to randomly introduce single amino acid mutations into the integrase protein on the top or the capsid protein on the bottom, and then measure the fitness of those viruses, as indicated by the black bars, and these little red dots indicate viruses that were completely dead. And you can see that it's very difficult, in fact, to find places in the capsid protein where mutations can be introduced and not be lethal to the virus. This would lead to the prediction that molecules that target the capsid protein would experience a very high genetic barrier to resistance. And this is probably because of the many functions that the capsid protein has to perform in the cell. In particular, during the assembly of the virus into immature and mature virus particles, there are two completely different hexameric lattices that have to be assembled, and they have to have precisely the right stability so that the virus can go through the molecular gymnastics that accompany the assembly and disassembly of the virus life cycle as it proceeds, as well as directing nuclear import. So capsid is I would argue, a very much underappreciated and under-targeted aspect of the virus life cycle that only recently has attracted the attention of the pharmaceutical industry. Now, in terms of our own work, what we recognized as a result of work on restriction factors is that they can be very important restrictors of host range. This, in general, is a good thing. Were it not for the fact that we have an ApoBec3 protein that cannot be targeted by the VIF proteins of all these simian immunodeficiency viruses, we would have been subjected to far more epidemics of AIDS viruses than we already have. This, on the other hand, is a bad thing because it means that HIV-1, the virus that we're mostly interested in, can only infect humans and closely related species. It cannot infect monkeys or any more distant species, and therefore that makes it quite difficult to generate animal models that are of great utility. That is not to downplay the significance of the simian immunodeficiency virus models, but we would very much like to have a model in which we can use the human virus to infect a non-human species. And so what we found a few years ago is that there are two key antiviral blocks that need to be overcome that would then allow HIV-1 to replicate in the cells of non-human primates, specifically macaques. That is the TRIM5 protein, which in humans is unable to recognize HIV-1, but in macaques is a devastatingly effective antiretroviral protein. Similarly, the HIV-1 VIF protein is very effective against human ApoBec3 proteins, but is nearly inactive against the equivalent ApoBec3 proteins found in macaques. But reasoning that we could engineer our way around these blocks, what we decided to do in collaboration with Jeff Lifson and his team a few years ago was to try infecting this species, pigtail macaques or Macaca nemestrina, a fairly frequently used primate species in biomedical research. What we and others found is that this species is unusual in having a TRIM5 protein that's ineffective against HIV-1. And so we reasoned that simply by changing the HIV-1 VIF gene from a simian immunodeficiency counterpart that can antagonize the ApoBec3 proteins in this species, we might be able to get an HIV-1 strain that could infect an animal. That indeed worked to some extent, but enabled an adaptation experiment that proceeded over a series of passages extending over years, where we took an engineered HIV-1 strain and then adapted it to replicate in pigtail macaques. So to facilitate the adaptation, what we did was to temporarily deplete CD8 cells from the periphery of these macaques during acute infection and then pass the virus from one macaque to another to another. And after around 4 passages, we could clone a virus, and at that point thereafter, so long as we CD8 depleted during infection, then the virus would cause AIDS in this non-human species. The first time this had been achieved for HIV-1 in a non-human or non-hominid species, I should say. So during this adaptation, many interesting things happened to the virus, one of which I'm showing here. So HIV-1 VPU, as I mentioned, acts as an antagonist of human tethering. It, as it turns out, we didn't know this at the beginning, but we do now, does not work against macaque tethering. But during the adaptation process, we found that the VPU protein mutated, acquired the biological activity that's present in humans and made it work in macaques, and now acts as a very efficient antagonist of the macaque tetherin protein. So what's interesting about this model is that we can manipulate to toggle between the two very extreme outcomes of HIV-1 infection in humans. If, as shown here in the blue and green colors, we administer the virus to macaques along with the temporary anti-CD8 antibody, we very reproducibly get rapid progression to AIDS with high viral loads and nearly complete CD4 depletion. If, on the other hand, we withhold the anti-CD8 antibody, then we get elite control. If we then later add the anti-CD8 antibody, after infection has been established, so simply displace the administration of the two manipulations in time, then we get a blip of virus, but then it comes under control again. This, to us, suggests that key battles are fought between the immune system and the virus during this very early phase of HIV infection that are effectively deterministic in the outcome of that infection, at least in this model, and perhaps more generally. And we can use this model, this is an earlier version of the model, to investigate interventions that couldn't be easily investigated in the SIV model. So this, for example, is a PrEP experiment where we have treated macaques with a three-drug combination that mimics a regimen that can be given to humans as a single tablet per day. We administered that combination for 2 weeks, and in the middle of that 2 weeks, we subjected the macaques to an extremely high-dose challenge, 2 million infectious units administered intravenously, and this particular PrEP intervention gave apparently sterilizing protection to that extremely high challenge dose. So that gives you a sense of what restriction factors are, what antiviral proteins and how they work, and a sense of how we might think about applying them to do something useful. And in the last few minutes, what I want to do is tell you something new that we found recently. It's essentially a new way that hosts have evolved to protect themselves against viruses. And in order to do that, this is an antiviral defense mechanism that's based on recognition of RNA sequences, based on their nucleotide composition that's targeted against non-self RNAs. So in order to do that, I have to tell you about a little bit of chemistry. So your DNA, all of our DNA is made of 4 nucleotides, A, G, C, and T. And those 4 nucleotides can be arranged in various sequences, and there are 16 possible dinucleotides, AG, AC, etc., etc. It turns out that of those 16 dinucleotides, one of them in particular, CG, is rarely present in our genomes. And the reason for that is here. Vertebrates have enzymes, DNA methyltransferases, that recognize CG dinucleotides and add a methyl group to that nucleotide to generate 5-methylcytosine. This is extremely frequent, particularly in mammals. About 70% of your CG dinucleotides are methylated. Why that's interesting is because the spontaneous deamination, which occurs at a low but non-zero spontaneous frequency, generates from 5-methylcytosine a thymine nucleotide. Now, this is not an unnatural base. This is another base that's found in your DNA. And so what happens when that occurs is that you get a mismatch, and your DNA repair machinery doesn't know which is the correct base and which is the incorrect base in the double-stranded DNA. And so sometimes it repairs the wrong base, and in so doing causes a mutation, effectively a cytosine to thymine mutation, particularly in the context of CG dinucleotides. And over the hundreds of millions of years that vertebrates have been in existence, this has resulted in a gradual purging of CG dinucleotides from the particularly mammalian genome, such that they're now present at about 1/5 the frequency that they would be expected to be there by chance. So there's no great mystery surrounding this process. Biologists have known about this process in vertebrate DNA for a number of years. What's been substantially more mysterious is the fact that RNA viruses that are not subject to methylation, mimic this process. So the RNA viruses that infect vertebrates typically have fewer than expected CG dinucleotides. And it's been very unclear why. That phenomenon is very prominent in HIV-1. So what you can see on this chart, the blue line shows the number of CG dinucleotides in a sliding 200 nucleotide window in random sequence, about 9,000 nucleotides of random sequence. Whereas in black, we see the number of CG dinucleotides that are present in the HIV-1 genome, and you can see that they have been dramatically suppressed. In red is an artificial mutant that we made, where we reintroduced CG dinucleotides into the HIV-1 genome. Taking advantage of the genetic code, we did not change any protein sequences, we just changed the nucleotide sequence. So what happens when we do that, and I'm showing this now for a particular segment of the envelope gene, although it doesn't have to be the envelope gene, it can be pretty much anywhere in the virus, is that the addition of synonymous, non-protein sequence changing CG dinucleotides is lethal to the virus. So here you can see the wild-type virus replicating in black, and then the virus where we introduced synonymous CG dinucleotides is completely dead. This is a specific property of CG dinucleotides, because if we make 3 times the number of other mutations, so synonymous mutations that don't make CG dinucleotides, the virus is fine. And if we make GC dinucleotides, just flip the order, again the virus is fine. So a specific property of CG dinucleotides. And what those CG dinucleotides do to the virus appears to be to destabilize their RNA. So what you can see in this slide are wild-type HIV-1 infected cells, and each one of these tiny red dots on the bottom picture is an individual RNA molecule. When we infect that cell with a virus that carries additional CG dinucleotides, you'll see that the cytoplasm of that cell has very few RNA molecules in. There are plenty in the nucleus. This little cluster here probably represents the site of proviral integration. That's not a specific property of this virus. We see that also in wild-type infected cells, in a proportion of them. But the key point is that RNA is lost from the cytoplasm of HIV-1 strains that carry excess CG dinucleotides. So what's responsible for this? It turns out that it is a known antiviral protein discovered in Steve Goff's lab a few years ago, ZAP. Zinc finger antiviral protein is known to inhibit the replication of a number of RNA viruses, but it wasn't known how or even whether this RNA degrading activity was specific for viral RNAs. What's shown on this slide is essentially a binding assay, a cross-linking assay, where represented on the x-axis is the HIV-1 genome, and on the y-axis is how frequently ZAP binds to that particular position on the HIV-1 genome. And you'll notice in the wild-type genome, it very, very rarely binds. In our mutant genome, it binds very frequently, particularly at positions where we have introduced CG dinucleotides. So here is the key experiment. If we take our HIV-1 strain that has these CG dinucleotides, it does not replicate at all in normal cells that have 2 copies of the ZAP gene. If, however, we use CRISPR editing to remove the ZAP protein from those cells, then this otherwise dead virus replicates just like wild-type virus. And then if we re-express the ZAP protein, then the virus is inhibited again. The wild-type virus that has its CGs strongly suppressed doesn't care how we manipulate ZAP. So what this, I think, tells us is that ZAP appears to be a component of a defense mechanism that recognizes non-self RNAs that have not undergone CG suppression. Of course, many RNA viruses have adapted to mammals, and in so doing, they have suppressed CGs themselves. But of course, for hundreds of millions of years, we have been exposed to viruses from invertebrates that do not have CG suppression through biting insects, and so this defense in part protects us against those viruses. In practical terms, what we think and actually know we can do is that introducing CG dinucleotides can provide a rational, tunable, so one CG dinucleotide has almost no effect, 40 CG dinucleotides are lethal to HIV. A difficult reverts mean to rationally attenuate RNA viruses. And of course, live attenuated vaccines, while not applicable in this field, are very much useful in studies of other RNA viruses. Okay, so let me summarize. If you've ignored the talk, now's the time to wake up for the 30-second summary. So you can go back and tell your colleagues what you heard. So, humans make a wide variety of antiviral proteins. And in order to colonize humans, viruses must evade or antagonize those antiviral proteins. Otherwise, they will not be successful. And what I hope I've convinced at least some of you of is that understanding the mechanisms by which these proteins work, how they're evaded, and how they're antagonized can provide opportunities to develop therapeutics, animal models, and potentially vaccines. Thank you very much for listening.
To introduce our second speaker, please welcome back Professor Françoise Barré-Sinoussi.
I'm very happy to introduce now Sandra Souza Ruby. Sandra is health economist with master's degrees in health economics from the Center for Economic Research from the University of York. For over 10 years, she has been an investigator at the Health Economic Unit at the National Health Institute in Mexico. Her research is directed at risk behaviors associated with HIV infection among men who have sex with men, MSM, and male sex workers in Mexico and Central America. Specifically, she studies the effect of the use of economic incentives in linkage to care and changes in sexual behavior among MSM and among MSW. Sandra is currently the principal investigator of the project development of a mobile program prototype for improving retention and adherence of HIV patients in Mexico City. These programs aims to improve the linkage to HIV care and HIV treatment adherence among MSM using mobile messages between physicians and patients. She is also a researcher for several projects that test the effectiveness of other behavioral incentives through multimedia technologies for MSM and the efficiency of the provision of HIV prevention intervention, including prevention to mother-to-child transmission, HIV testing and counseling, and male circumcision in sub-Saharan Africa. In 2011 and 2013, Souza is assisted in developing a national survey to estimate the HIV prevalence and risk behaviors among MSM in Mexico in order to evaluate HIV prevention intervention funded by the Global Fund to fight AIDS, tuberculosis, and malaria. Please welcome Sandra.
Good morning. I'm here to tell you about the humanitarian crisis in Venezuela and how this crisis is affecting people with HIV. My name is Jesus Aguais. I was born and raised in Caracas, the city where I was diagnosed with HIV 29 years ago, a time where HIV diagnosis was a death sentence. I got lucky thanks to the antivirals and the fact that I moved to the US, that I'm still here. Now, almost 30 years later, my fellow Venezuelans with HIV are facing the same reality. For them, their HIV diagnosis is also a death sentence. The International AIDS Conference is mainly focused on following up on the progress of achieving the 1990 goals. However, data from the 2017 report presented by UNAIDS here shows that Venezuela has only reached 7% of viral suppression, the worst country in Latin America and among the 10 worst countries in the world. Venezuela has been left behind. Neither donors nor the Global Fund wants to help. In the meantime, there's 120,000 people reported living with HIV in the country. And the real number, it might be close to 300,000. There are at least 11,000 new infections a year. And in 1999, 1,242 people died of AIDS-related complications. In 2015, that number doubled. Stockouts of ARVs has been going on for a month, and 71,000 people are directly affected today, with 70 to 90% of treatment absent from the distribution chain. 80% of people living with HIV today are without treatment in Venezuela. There's no viral load test. There's no HIV rapid test. There's no condoms. There's no loops. Women with HIV are not able to prevent mother-to-child transmission. They have to choose whether to breastfeed their babies and transmit HIV or let them starve to death. Our indigenous community, the Waraos, in 2013, 10% of the population was diagnosed with HIV, and they did nothing about it. There's no rapid test to know what's going on, and it might be 20 or more the percentage of population living with HIV in this indigenous community. So we call on all of you. We call on urgent global solidarity. We need to get treatment to the people in Venezuela that are dying. Gilead, Viv, Roche, just to name a few pharmaceutical companies, has the ability to donate ARVs today. The people of Venezuela need CD4 and viral load diagnostic tools. They need rapid tests today. I urge you to join us sending this message to all pharmaceutical companies and to the international community. We know how to get the much needed help to the hands of the people in Venezuela. Please support us. We have flyers for more information. Please act now. Thank you.
To introduce our final speaker, please welcome Ambassador Lambert Grijns, Ministry of Foreign Affairs, Netherlands. Lambert Grijns is Director of the Social Development Department and Ambassador for Sexual and Reproductive Health and Rights and HIV and AIDS at the Ministry of Foreign Affairs of the Netherlands.
Good morning. It's my great pleasure and honor to introduce to you Dr. Nadia Sam-Agoudou. She has a research interest in HIV and young people, a subject which is close to my heart and which will actually also be high on the agenda of next year's International AIDS Conference in Amsterdam. And today, Nadia will speak about what it takes to end pediatric HIV. Dr. Nadia Sam-Agoudou is a senior technical advisor for the Pediatric and Adolescent HIV program at the Institute of Human Virology, Nigeria, and the Institute of Human Virology at the University of Maryland School of Medicine, where she is also assistant professor of pediatrics on immunology and infectious diseases. Nadia has focused her career on the control and elimination of infectious diseases of public health importance among children. And she was the 2007 American Society of Tropical Medicine and Hygiene Honorable Mention Young Investigator for her work on pediatric malaria in Uganda. And she has further research experience in Ghana and particularly in Nigeria, where she has provided technical and policy-making support to HIV programs since 2010. Her experiences in supporting Global Fund, PEPFAR, and nationally funded HIV programs drive the formulation of research questions for further study. And her research interest is in using implementation science methods to generate evidence for sustainable HIV prevention and control among children and adolescents in resource-limited settings. She served as a principal investigator for one of the 6 World Health Organization-supported INSPIRE PMTCT studies, and she's currently a principal investigator on a US National Institute of Health-funded study on healthcare transitioning for adolescents living with HIV in Nigeria. Dr. Sam-Agoudou obtained her medical degree from the Mayo Medical School and completed her residency in pediatrics and a fellowship in pediatric infectious diseases at the University of Minnesota. Nadia, you have the floor.
So thank you so much for the introduction, and bonjour and good morning to everyone. When I first noted the topic I was supposed to discuss today, my initial reaction was, that is quite a wide topic to treat, ending pediatric HIV. But we've come a long way from a time when people didn't even believe that children could have HIV, unfortunately. And now we have knowledge on how to prevent it and actually to end it. And therefore, today I'd like to share with you a few lessons from the global initiatives to prevent and now to end pediatric HIV. So first of all, I'll do a sort of state of the PMTCT overview. How far have we come so far in PMTCT? I will talk about the PMTCT toolkit for success. What are the essential elements of our toolkit for our success so far? And then I'll discuss the global plan successes and challenges. And then also note that the discussion on global plan in terms of successes and challenges is to some extent an indicator of what is going on in other program areas, for example, treatment as prevention among non-pregnant adults and children. And then finally, we will touch on next steps towards ending pediatric AIDS. So over time, we've had increasing evidence that triple drug regimens given for longer periods of time to HIV-infected mothers is more potent or are more potent for preventing vertical transmission. And so over time, the WHO guidelines have evolved from moving from option A to option B, and then to option B versus option B+, and now going on towards treatment for all for life. So this graph shows how an increase in access to antiretroviral drugs has really gone towards success towards preventing vertical transmission. Now you see that there are different regions of the world that are doing well with regard to access to antiretroviral drugs. For example, Eastern and Southern Africa, the Latin America and the Caribbean, but there are other areas of the world that continue to have challenges with increasing access to potent drugs for pregnant women living with HIV. And that includes Middle East and North Africa, East Asia and the Pacific, South Asia, and West and Central Africa, where I was born and where I live and work. Now, when we focus on the 21 sub-Saharan African countries that are the focus of the global plan, this graph is really making a simple point, that as access to potent drugs has increased among pregnant women, new infections among children has decreased. And with the introduction of the global plan in 2011, access and therefore drop in child HIV infections has gone towards our goal to end pediatric HIV. Now, the key toolkit items for PMTCT so far, in terms of our success so far, have been what? For example, we've had policy that is supporting expanded ART eligibility and duration of treatment for pregnant women. We have drugs, supply of drugs, and access to these potent regimens. We have testing, increased access to testing and coverage with rapid tests. We have human resources for health and service delivery, where we are decentralizing delivery of services and having consistent access to the interventions that we know are working. We have enhanced monitoring and evaluation systems, which are critical to track our progress and the quality of the services we are delivering towards the outcomes that are desired. We also have community engagement by way of community engagement of people living with HIV, as well as community engagement in terms of geographical engagement of communities in which people living with HIV, whether they know or do not know their status, are living. And we are maximizing advocacy and participation with these groups of individuals. Now, we cannot talk about ending pediatric AIDS without talking about the global plan and its achievements. The global plan prioritized 22 countries for the elimination of mother-to-child transmission, or we can say for the elimination of parent-to-child transmission or vertical transmission. The main objectives for the global plan were to reduce new child infections by 90% and provide treatment to at least 90% of pregnant women living with HIV, and also to reduce the mother-to-child HIV transmission rate to less than 5% among breastfeeding infants and to less than 2% among non-breastfeeding infants. Now, the global plan report shows that there are essentially three groups of countries in terms of its final report. We have countries that I call the high achievers, countries that are the middle achievers, and countries that were the low achievers. And for now, let's focus on the countries that are the high achievers. And these are countries that are nearing the goal of 90% decline for HIV infections among children in the global plan evaluation period. And we have these countries including Burundi, Malawi, Mozambique, Namibia, South Africa, Swaziland, Uganda, United Republic of Tanzania, and Zambia. Now, we also have to mention the countries that have actually been validated as eliminating mother-to-child transmission of HIV as well as syphilis. And these include countries like Cuba, that was first validated in 2015, and then following that, Belarus, Moldova, Armenia, and Thailand in 2016. So you ask yourself, what is the secret of these countries, these high achievers and the ones who have achieved validated elimination? They've had strong policy that is backed by sustained political will, even across governments. They have strong or strengthened health systems. They have functional monitoring and evaluation systems. They've conducted operational research in order to drive their reorganization of their priorities. They've had adequate and sustained funding, and they have strong advocacy and community participation. Now, the point here is that although we know what the PMTCT toolkit is, the essential elements, the toolkit from country to country might be slightly different based on the context of the HIV epidemic in that particular country. So now let's move on to looking at the other categories of countries from the Global Plan performance report. Unfortunately, where I live and work, Nigeria, is in the very bottom, along with Angola. But what I also want to note that when you combine the countries that are middle achievers with the countries that are low achievers, you have about 11 countries. And what I also want to point further is that out of these 11 countries, 6 of them are in West or Central Africa, And 5 of these 6, when you rank all the global plan countries, are in the bottom third. So what is happening in these pediatric AIDS hotspots? We have inequitable funding for the HIV response. Not lack of funding in some countries, but really inequitable. We have weaker health systems. We've had conflict, instability, and competing interests, which to some degree contribute to the weak health systems. We have relatively lower HIV prevalence, which you might think makes it easier to deal with the epidemic. But however, there are more resources needed to find, test, and treat people in these low prevalence areas, particularly when you have a large base population in which to find these individuals, such as it is in Nigeria. And to some degree, we have to think whether our sense of urgency is maybe not as high as it should be because we have a low prevalence. So points I would like us all to consider. There are things that I call the hardware, the PMTCT hardware, which are the drug regimens, the test kits, funding, numbers of staff needed to do the work. And then we have the software, which are the behavioral drivers of success. However, we have 2 categories of issues with the behavioral drivers, as I see it. Issues 1 are largely among clients. We have stigma, we have discrimination, denial, lack of education. And then issues 2, which are among people like myself, who are healthcare workers, who are policymakers, who work for the ministries of health in the different countries. And I really think it is time that we galvanize our efforts and put more personal and professional value on the hardware that we need to get to the people that need it. So what we need, though, is impactful behavioral interventions. In addition to what we already have, we have to have sustained uptake and desired outcomes, especially now that we are treating all for life. And we need to do this in combination with the hardware that we already have and that we know works. So the INSPIRE group of countries were 3 countries, Malawi, Nigeria, and Zimbabwe, that had 6 projects funded for PMTCT. And all of these projects were looking at retention. I'm going to highlight 3 projects that were looking particularly at peer support in different forms. And peer support meaning that support provided to other HIV-positive women, from women who are HIV-positive, who have personal experience in PMTCT and are trained to provide psychosocial support to other HIV-positive women towards retention and uptake of services. Now, for the first study, we have Sampiri and his team who were implementing this study under Option B Plus in Malawi. They had 21 sites, over 1,000 women, and we have a standard of care where there was no peer support compared to facility-based support, which was also compared to community-based peer support. This was a 3-arm study. And for 24-month retention, the results were that there was 66, 80, and 83% retention, and this was statistically significant. Now, for the second study, which was in Nigeria, by myself and my team, we operated under Option B for this study, and we had 20 sites, all in rural areas and at the primary healthcare facility level. We had almost 500 women, and we compared unstructured versus structured peer support. And by structured peer support, I mean daily supervision, having a logbook, a standardized logbook for mental mothers to document their interactions with clients. We had periodic evaluations of the mental mothers' activity. And then for 6-month retention, what we found was that there was 25% retention in the unstructured arm versus 62% retention of HIV-positive women in the structured peer support arms. For the last study, which I will talk about, this was in Zimbabwe with Jeff Foster and his team, and they operated under Option B+, looking at 30 sites and 350 women with standard of care with no support at all, comparing that with facility-based mother support groups. So this was a different kind of peer support in that it was a group support. And for 12-month retention, women who were categorized as being regular attenders to support group versus non-regular attenders were 3 times more likely to be retained. So that all being said, what are the next steps post-global plan? And the post-global plan is an acceleration plan. We need politicians to champion political will. We don't simply need political will, we need politicians to champion political will. We need to increase allocation of funds towards vertical transmission prevention and elimination from the domestic funding. We need to mobilize people living with HIV in the acceleration plan. The studies I showed in the prior slide show how well peer support can work in improving PMTCT outcomes towards the elimination of vertical transmission. And when we mobilize people living with HIV, we are able to have better community education, and we are able to optimize the so-called software strategies, the behavioral drivers that should be driving our success, in addition with the hardware that we have. And then we need to target our interventions to the right places. We need multi-component interventions. So the interventions of peer support and behavioral interventions I mentioned previously are combined with all the other interventions that have been proven to work with PMTCT. We need to have sub-national level data and cohort-based data so that we know exactly what we are achieving, where we are achieving it, and where the gaps are at the country level. So I will leave you with this quote from the UNICEF website showing that there's been great progress in reducing new HIV infections among children, but it is not fast enough, which is why we need acceleration. When we have the tools in hand that we know work towards our success, and yet we continue to fail to meet our goals, that is a missed opportunity. And we cannot afford to have any more missed opportunities for eliminating pediatric HIV. So I'll stop there and say thank you. I'd like to thank the International AIDS Society, particularly Linda Gale and Landon Meyer, UNICEF, Geneva and Nigeria, particularly Dr. Dorothy Mborengacha, UNAIDS, Karusa Kiragu, and WHO Geneva and WHO Afro, Dr. George Sivery from OGAC, the Institute of Human Virology, Nigeria and Maryland, where I work, and really to all people living with HIV in Nigeria with whom I've worked and for whom I've worked over the past few years. Thank you very much.
The conference organizers would like to thank all the speakers and co-chairs for their participation. The plenary session is now over. We wish you all an excellent day at the IAS 2017 conference.